Hello, welcome to
Hi,
yeah, welcome to Microbiology Journal Club, where we nerd out big about all things small.
my name is Danny, and in a previous life, I dropped out for my PhD in microbiology at the University of Chicago, where I was infecting skin that I grew with MRSA. Nowadays, I'm a fact checker for pharmaceutical advertisers and the president of Biotech Without Borders, a nonprofit based in NYC dedicated to enabling the public with the tools of biotech.
My name is Faz. I have a PhD in microbiology, and I've mostly worked with bacteria. But since leaving academia, I've worked with as a research integrity specialist, and I'm now currently working as an editor of a science journal.
Yeah, every week we meet to talk about microbiology. Today is our news week, where we do an overview of papers that we have seen in other news or other sources, and we try to find an article that we want to cover next week in more detail.
Next week will be our deep dive week, where we take the article that we choose today and we look at it figure by figure to learn what it can and cannot tell us about what it's talking about. So make sure to subscribe to satisfy your microbiology curiosities.
And you can follow along with all the papers that we discuss on either week in our shared Zotero library linked in the doobly-doo below.
Yeah, and we want to hear from you, so please use the comments or tweet us using the hashtag microTWAC hashtag. Um,
Nice.
yeah, so now let's get into today's news because there is some big news in the UK. So on Friday, our Prime Minister came up and announced that this new B117 strain is well, they found evidence that it could be more severe, that it could be more deadly potentially than the original recipe
Spooky.
strain. So it's I mean, it's kind of a panic-inducing thing. So I know that there are some people out there who are kind of very frightened right now. So I decided to go in to try and look into the details, try and figure out what actually is the evidence that is out there. And there isn't really that
Yeah.
much
Yeah.
at the moment, unfortunately. But I will present because again, this has been uh, almost like urgent. They've organization
Mhm.
that is here to check out the responses to to various like outbreaks, and so that's basically gonna be my primary source for what I'm gonna be talking.
Yeah. We've covered some of their. I think I mean this new variant emergence has been going on now for maybe like a month now. I would say in the news, and we've pulled on this group's papers before, right? They've had technical briefings that have let us know like that the variant is out. Um, so this is just from this group.
Yeah, so
Um,
so before going, so if you wanted to download papers yourself, we've got all of our like sources in the description, so you can look at these things. And so they're talking about this. They call this strain SGTF, which is a new acronym for the strain because we've got at least three so far. There's B117, there's
yeah.
VOC 2020 12-01, and now SGTF, which stands for S gene target failure, which basically is how we're currently detecting it. Um, so we've
Yeah.
talked about in previous shows that the spike protein can't be sequenced. Well, it's very difficult to sequence in this, so often drops out of tests. So when you
Yeah.
get
There's a mutation, right, that deletes one of the regions that's used for the PCR test.
yeah, the H69V70 deletion, which causes
Right.
this S gene target failure, which is actually kind of convenient for testing purposes because that means that we can actually see we're already testing for multiple genes. So research labs have
Mhm.
come back and found that some of these have dropouts, and then they've used that to kind of proxy where the P117 is infecting. Um,
Yeah.
and this also so they've had hints of it being more severe. So there's been a previous report where they found that the 28-day case fatality was assessed, uh, and like say out of the variant cases, 12 out of 1340 were had people dying within the 20 days, compared to 10 of the similar number of wild type cases. So there has been this
Hmm.
sense that it could potentially, but there hasn't. They haven't had enough results to get that statistical significance. Um, so
So that's in the SAGE meeting paper?
this is in a pre. So yeah, I did dig up this one because there was a reference in the Sage meeting paper. So I looked in
Mm.
through. So the title of this one is can put it through investigation of novel SARS-CoV-2 variant of concern 2012/01 technical briefing 2. This was released in like I think late December.
This, yeah.
So yeah, I so This builds on that. So what essentially has happened is that since then, people have gone out in from the universities and they've been collecting data on this. And on the 15th of January, two papers were presented to this group describing and things. We don't actually get access to these papers. So these papers are not published yet. They haven't even been submitted to a pre-publication server. So the only evidence I have their existence is from this meeting paper. Um,
Hmm.
so hopefully, like when in the future when they're finished up, they'll kind of come out and like we can actually read through them properly. Um,
Right.
but in this case, we're just getting a summary of these papers because essentially these
Oh
researchers.
yeah, that's the Ferguson. I'm just looking at the references on the Sage paper now. That's the Ferguson 2021, right? And the Davies
Yeah, that's
2021
right. So
unpublished analyses. That first author, Davies, that was the one that we read last week,
yeah,
actually,
that's
right? That's the
right.
same first author as the modeling paper.
Yeah, so it's from the same group. That's from the London School of Hygiene and Tropical Medicine, and Neil Ferguson is
Mm,
from Imperial College. So they've been involved with the government response, um, and they've been taking in data from the testing. So testing of key workers and people in social care and other sectors, and they've been taking in data. And so I'm gonna so there's firstly these first two papers I'm going to focus in on a bit. So the first paper was based on 1.2 million tested individuals, and they found that they looked at like 2,583 deaths, and of which of those 384 were from B117, and they checked like kind of compared like the case fatality ratios for these two. So the case fatality ratios is the probability of you getting a fatality based on like like the proportion of the population who has that strain who will die from it. Uh,
yeah.
so and then they don't present the absolute numbers. What they present is a comparison. So they essentially say, well, take one of those numbers and divide it by the other to get. So they say that okay, com- so they kind of say relatively to the number of cases, there's about 35% more people dying in the B117 group, which
Hmm. Interesting.
so yeah, they try to look at
Yeah, so they make like it's a ratio, right?
yeah
Like between the two groups to see that one might be more. But like that's one of those things because it's done in this what retrospective,
yeah
right? Type data, then there's all those different covariates that have to be controlled for. And I guess without seeing the paper, we don't really know how well those things were controlled for. There could be other reasons for that.
yeah. So they do try. So in this report, they do try to mention some of the covariates that there's proportion of beds capable. So looking at kind of whether this is due to the healthcare system being overwhelmed and losing
Right.
staff in critical
Yes.
care. See, like that doesn't because sometimes that can happen in areas that are already having a high number of cases, and those
Right.
areas that are already having a high number of cases, also have this new strain coming up. So those two things
Hmm.
can be linked, but they're just trying to pull it apart, seeing whether if they're not linked, then maybe could that explain it? And they,
Yeah, yeah. I mean, that's good. I mean, we saw this before. Like, you want to be removing, this is this is the same group, right? Like, if they're doing a model of some sort, you just want to remove hypotheses, right, as to like what it isn't, and then hopefully you're left with what it could be.
yeah, excellent. The Imperial College also has a paper that was essentially taking uh, data from the same cohort. So they're all taking data from
Hmm.
this like pillar two testing, which I'm not going to really go into. It's not really that important to understanding what, but essentially it's like the same type of data, and they use different models. So they use different methods to analyze the data, and they also come up with a ratio of about 1.36 or 1.29 depending on which model they use. So that's still uh, kind of an expansion in numbers of the B117. So
Sure.
and then they talk about Public Health England also is presenting data, um, again coming from a similar source, well the same source where they looked at like the odds of a B117 case going to hospital, and they say that it doesn't look significantly different, but there is a similar increase in the ratio of deaths. So they're still coming up with a 28% increase in deaths. Um, so these studies are operating on the same data, so there might be so it's not necessarily too surprising that they're coming up to the same conclusion because if there is that difference in the data, and it does strengthen the idea that B107 could potentially be be more severe. Um,
Yeah.
but there are some things that do need to be considered. So they do mention that some places might not be reporting all of their B117 cases because of the way it's detected. So we mentioned it's detected by spike gene dropout. Thing is, like
Hmm.
when you've got like you're running a PCR test and you've got a low amount of virus anyway, genes do tend to drop out anyway, just because of the way that the test is going. So some labs have been trying to set a baseline of like if any like kind of they spot any gene dropouts under a certain level, then that could just be because there's so little virus there. So that
Hmm.
in a sense,
Interesting.
so they theorize that that could be a cause of a confounder. That could be why the B117 cases could potentially seem like they have a higher viral load. It's because some of these labs aren't reporting
Mm,
on the cases that have a lower viral load. Um,
sure. Yeah, I mean, I was gonna say too that I don't think that the diagnostic, like this S gene dropout observation, that was made. It wasn't like the diagnostics were designed to detect
no,
this, right? It's just like it so happened that they started detecting these things, and now it's like, oh, this information may actually be useful in monitoring the spread of this variant, but it's not intentioned for that, so the right controls might not be around, right? I think is
yeah,
one of the things that statement is kind of like the right controls might not be around 100%.
yeah. So in order
Yeah.
to remedy this, they basically set the same baseline for both studies. So they said, okay, if we're going to treat the B117 like this, we have to treat the other data like that. And because
Mm.
of that, they looked at and they found that there was still that increased severity, that increased death rate.
Oh, nice.
So
Yeah.
um, uh,
So that's like truncating. They like truncated the data.
yeah,
Um, yeah.
but there's still this mystery that the cases they didn't see an increase in hospitalization, which is what you'd kind of expect to see if the infections more is greater. What they saw was just an increase in death. So that's kind of a mystery, and it's not something that we can
Mm.
really delve into until we have more data in and more of this stuff has been
Yeah.
peer reviewed.
Yeah.
Um, so from a scientific perspective, there are still loads of questions about what is happening with the fatality rate. There is a lot of uncertainty here, and I can sort of see, like, from a decision making perspective, why they decided to announce it because they want people to know, and they also want people to stick by the lockdowns.
Um, I mean, this is one of those one of those instances again where it's like you have to make some sort of policy decision, some sort of um, public facing statement before you have the whole idea. But it's reinforcing that with these lockdown measures, as people get kind of tired about doing them, it's still really important to do because there are everyone is working together to reduce the spread of this virus and like this variant too,
Mm.
right? Like, so with the unknown, better to err on the side of caution and just stay indoors if you can, right? I mean, that's always the big caveat, right? People have to also make their living.
Yeah, so I
Yeah.
and there isn't really so even though we've got this new news as terrible disease, there's not really much we're going to be doing differently though. So that's the thing
Yeah,
that
yeah.
so it's just basically
Just continue on the way it's been going.
yeah. So it's
But if you haven't been, right? Like I think that to me it's always like all of these like increased lockdowns and whatever, it's really saying if you haven't been doing these things because you thought it wasn't so bad, like it really is quite bad. I think you should be paying attention to your local regulations.
yeah. So on that note, I think we're going to start to the actual scientific papers that have been
Hmm.
looked around, and I think the first one we're going to take a look at is the impact of SARS-CoV-2 B117 spike variant on neutralization potency of sera from individuals vaccinated with the Pfizer vaccine, BNT162b2. So
Yeah.
yeah, we covered a paper on this previously, but that had very low numbers of
Mhm.
participants. This one's slightly bigger. Um, and because of that, so this one has 23 participants who were vaccinated 3 weeks prior. So these are people who were vaccinated, but they only got their first shot.
Mm.
And
Yeah, and they take their serum out and then they test it on pseudotype virus,
yep, pseudotype
or is
virus.
it real
I think they.
pseudotype
So
virus?
yeah, they created their own version of the spike mutation, the spike protein. First version they had like 3 of the crucial mutations, and the second version they had all of the crucial mutations, B117. So hopefully that's kind of
Hmm.
so they can hone in on what the actual reason for the the causes. Um,
Right.
so the one thing I will note is that the median age of the group was 82, which is, I think, quite a good thing because that's most of the people who've been vaccinated at this point. But also means that
Mm.
there is a lot of variability on that data because in that 80. So I'm gonna see if I can pull up just one.
Oh, like some individuals don't get the same immune response as others, is what you're saying.
Yeah,
Old immune systems are maybe a bit slow to pick up.
yeah. I think this illustrated best on like Figure 1B. I don't know, so I don't want to like go into too much detail like reviewing this, but Figure 1B has this L-shaped graph where you've got like this response to spike. So they see how well their serum responds to spike, and you've got this age and their response to spike. So from like so at ages 20, there's like 20 to about 60, they've all got this response to spike, and then there's like this
Hmm.
massive like kind of
Oh
line
yeah.
over
It's very
80.
variable.
Yeah. So that's and this is just like looking at the age responses to the vaccine. When they once they go testing to the B117, they find that there is a reduced ability for it to neutralize for serum to neutralize the B117 spike. Um,
Hmm.
so the idea is like it they still get neutralized, but it just takes more antibodies to do it.
Hmm.
So
It's
yeah.
interesting. I mean, it's hard to say, right? Because these are all taking it in vitro. Like, I'm like trying to think of how does that translate to like in the body,
Mm,
right? Because it's like this is serum that they took out, and they're trying different concentrations of it. And right, like in the body, like it's not pure neutralizing
mm,
power that the how the serum works, right? There's other cells that recognize when stuff has antibodies stuck to it and do
yeah.
things. Um, so yeah, it's hard to say
Yeah.
what they're telling us here. I mean, it's supposed to be preliminary information to say, don't worry, like your antibodies are still recognizing at some level these viruses. But yeah, the sort of the significance biologically, um, I couldn't tell you
Yeah,
from this.
I completely understand that because for a lot of these vaccine studies, we are using serum as a proxy for the whole immunity, but we know that immunity is much more complicated, complex and diverse than that. Um,
Yeah.
so
Yeah. I mean, this is like one of those things where it's like, this was how they did their this is how they did their phase one studies, right? To show us that, oh, neutralizing antibodies made. This means we think it's going to be effective, but you still don't know if it's effective until you do that phase three study and you see how the efficacy is going. Um,
yeah,
yeah, I mean, maybe there's like, if there was like an animal, I almost want to say like this would be great for like an animal model of some sort, to help support their point.
yeah. I mean, it's going to be a bit, so we're going to go into that a little bit later, but I think it's difficult to find an animal model at the moment because well, it depends because we've already got 501Y mice, but they're not necessarily going to be representative of that disease in humans because that was the
Yeah.
threshold for it to be infectious in mice. Um,
Right. Right. Right.
but yeah, and this is one of those papers where the difference is significant, but not necessarily clinically significant. They've
Yeah.
so there's lots of stars, but the actual like kind of averages and confidence intervals overlap quite a lot. So
Mm.
feel like it's important to note that there is a difference. Um,
Yeah.
so the next paper we'll be looking at is mRNA vaccine elicited antibodies to SARS-CoV-2 and circulating variants. So this is interesting because this tests both the Moderna and the Pfizer, like,
Hmm.
and
A side by side.
yeah, well, it's almost side by side, but except they had 14 people who had got the Moderna vaccine and 6 people had the BioNTech. So it feels like more or less like side by side, and more that they just decided to put everyone in the same pool because they didn't have enough people
Right.
in either
To
one group.
study.
Yeah.
Yeah.
Um, so again, this is another pseudotype virus. So
Same where they take plasma out and see if it's neutralizing
yeah,
things.
basically, this one goes a little bit further, I think, because uh, so because again, it finds more antibodies need to be.
It's trying to oh,
No,
sorry. Go
you go
ahead.
first. I
I was just scrolling through figure two. It's trying to define like the antibodies that are being made specifically,
Yep.
like how they map back to certain B cell populations, it seems.
Yeah, exactly. And that's what I find really interesting about this paper because this goes out extra and does like cryo EM to map where those antibodies bind to spike protein and how those.
Ah, that's nice.
So it goes into a little bit more detail. Now I quite like that having the extra detail. Um,
Yeah. Well, I mean, this is one of those. This is one of the pieces of information that dovetails well with other information about like convalescent serum,
Mm,
right? Because I think people have seen in convalescent serum, you have this data like, oh, here's where your antibodies will bind to, or
mm.
whatnot. And then this is kind of like, okay, well, what about in vaccinated serum, right? Which is not convalescent serum. The immunity got formed in a different way. Yeah, this could sort of teach you of like, oh yeah, this vaccine is giving us maybe novel epitopes, right, to recognize versus a native infection.
Yeah, and the thing that's also quite cool is this one also covers not just the B117 variant, but also the variant that's been seen in South Africa recently. Um,
Mhm.
so that's
Or rather, like it speaks to the
yes,
mutations that have, yeah.
yeah,
Because these people that they take the serum from, they're not they didn't get the variant infection. It's unknown what infection they
yeah.
got, right? Yeah.
Well, I mean, these people
Um,
got the
or
doses of vaccine.
right, they don't even get infected.
Yeah.
It's just this is just yeah, vaccine serum. Okay. Hmm. Yeah, interesting.
Yeah. So next paper is going to focus a lot more. So this is on the South African variant specifically. Again, like so, the last three papers we've done have all not been peer reviewed, by the way. So I think that's important to note, so that you guys don't get too frightened about this one, because this is a paper that is almost designed to make me frightened. Um, because they looked at
Oh yeah,
yeah,
escape.
it gets really like kind of frightening. So firstly, they looked at like current monoclonal antibody treatments, and they showed a decreased neutralizing activity against South African variant, specifically linking it to the K417N mutation and the E484K mutations. Um, and they have these like kind of scary graphs where like things come up in black, and it's like I'm not going to show on the screen, but because I but yeah, they also showed it has like decreased neutralizing activity in convalescent serum, with nearly half having no detectable neutralizing activity against the new strain. So
hmm
yeah, so it's
Again, pseudotyped. Is this pseudotype
I
virus they're
believe that
using?
they are using a pseudotype virus. Let me. Okay, I guess I'm gonna be pulling this paper up to show people.
No, they do actually a neutralization assay with live virus.
Mm.
Um,
So
yeah, interesting. It's a short paper
yeah,
actually.
it's very short, and uh, yeah, I didn't wasn't really able to get my teeth into like how they represented their data. Um, but I mean, what the outcomes seem like kind of it seems like a kind of scary paper to read. So um,
Yeah, sure. I mean, the title says that it escapes, but again, this is where it's like, okay, so it escapes or it doesn't escape. What is the impact actually? Like, if we're fusing this with the BioNTech
mm,
information, right? Where it's like, oh, well, then if you get this vaccine, then maybe, and this is convalescent.
the,
Oh, yeah, right.
so
So,
yeah,
like, I mean, this is another really interesting thing. It's like, I mean, I would love to see when this gets shaken out more, right? Like, is convalescent immunity different than than vaccine immunity, right?
yeah,
Meaningfully for people, because remember, convalescent plasma was approved and used to some success
hmm
as a therapeutic. So, like, there's something to that
yeah.
that convalescent serum is going to work in some ways, but I guess it is maybe different in format um, to immune serum, and maybe that means it's not as effective. But again, unknown, right? Like this is just what people are throwing out there. These are the hypotheses people are trying to support with data, and there's just yeah, there's still not enough data. Such we have to really read a lot more to be able to come out with a Coles notes for this.
Yeah. So this is one of those. Yeah, I mean, this is something I'll be keeping an eye on a bit more for this story, because I actually there were like two other like papers that were saying the same thing, but even shorter like papers which were saying
Yeah.
that okay, we tested out, and this convalescent serum doesn't seem to neutralize the South African variant very much,
Hmm.
but
We haven't really talked about the South African variant, right? It's is it also an S gene dropout, or is it
it's
just I think it has 501, right? It has
yeah,
501Y.
it has 501Y and has a couple of other mutations. I think it might be an SG dropout. I'm not sure. I don't think so. So I don't think so. I think that was the thing that defined the B107. This one is I think called B15. I need to look that up before I
Sure,
say it
sure.
completely wrong. Um,
But I mean, I guess that maybe like what the reason I asked that question is just to like bring that clarity for people who are listening
mm,
along, right? Is that like these right? And we said it last week. I guess it never hurts to repeat over and over again. Is that like the mutations are all over the place, right?
mm,
Like there are a set of key mutations that we think might be driving the phenomena that we see, right? And that's where things have been focused. But then there are other ones. They could be helping out. They could be inhibiting. Yeah, and I guess this is like one of the reasons why travel, I guess, is still something that should be pretty touchy because you didn't want you. It's one thing to have these variants like occur through natural selection in
mm.
their different localities, and then another to also like be moving them moving them around to do the maybe like the airplane assisted natural selection where like we see what's most successful around the whole world.
Yeah. Um, so yeah, that's that part of the scare thing. So that's the current news. So that's what's currently frightening me because I'm now looking up the South African variant a lot more and just digging up papers.
Yeah.
But the thing is, there are lots of low quality papers coming out, and it's quite hard to pick apart. So
Right.
there's
And I think yeah, especially because like all these mutate these variants are like clusters of mutations. So like it's easy in the lab to make some of those changes,
hmm,
like or like to introduce one or two of those, but then that's not the whole thing. It's not the live virus. It's like the model. Yeah, so it's pretty tricky. But I mean, I think we still feel pretty confident after reading this stuff that there is some sort of there's a high likelihood that there's some sort of transmissibility, no, viral titer thing going on,
yeah.
right? That like these are growing higher in people. And so they do warrant like a little bit of extra care. I mean, the same care we've been taking for months now, but like extra care nonetheless.
Yeah, definitely. And I think we're going to keep on the kind of theme of immunity and
Hmm.
by looking at
Oh,
the evolution of antibody immunity to SARS-CoV-2. Um,
hmm. So this is actually on the other, this is like evolution on the human
yeah,
side, I think is what they're getting at here. And they just want to see like, so I think we've spoken to this point before where um, your B cells, after they get clued in that this is like a trouble antigen, they will undergo their own hypermutation to iterate on like several different variants of the antibody that worked best or that neutralized or not neutralized that recognized the danger antigen. And so this is speaking to that, right? That like, oh, at the beginning you're going to be making one set of antibodies, but let's see like after you've settled down into a certain type of memory, what antibodies come up next when you're challenged. I don't know if they challenge people
uh,
though. Uh,
they sort of they do, there's this quite interesting where they take some sampling and they look at asymptomatic individuals and they check to see
mhm.
like whether they still have some kind of antigens of COVID, and they found that some of them do keep antigens around, and that help, and they theorize that that's what's
Or keep antibodies that recognize those antigens around, right?
yeah, they actually like look at like some parts of the gut and they seem to like test for the antigens, which is kind of odd.
Mm,
And it's so
yeah.
I felt like that part of the paper would need a lot of expansion, but um,
I see. It's figure five, right?
yeah.
Where it's like something about their story is linking back to like where the antigens might be hanging out, I guess, that like keep priming your immune system to make a certain type of
Well,
antibody.
it's interesting because they show that like memory B cells stick around, so the antibodies responses go down, but the B cells stay and they still evolve. So the actual like
Yeah.
antibodies they create are still mutating, still developing to become better. And so they theorize that that could be one of the reasons. Which,
But that's for I think that's true for all
yeah,
infections that you have that cell compartment do those things.
yeah.
So right, like I sort of see this as like a oh, okay, like this is how our body's dealing with this specific infection. But yeah, it is interesting. I like that we're just diving into the B cell part. And now that we have different ways of eliciting B cell immunity, right, vaccine and accidentally getting infected, then it would be nice to know a bit more on that
I mean,
topic.
the thing that I'd like to see is a T cell version of this article because T cells are such a massive part of the antiviral immune response, and that
Yeah,
they really they aren't given enough credit. And so all the papers we looked
right.
so far looked at the antibody response, but that's not the primary antiviral response. That's more for like things like bacteria, but antiviral responses is
Yeah,
focused.
where it's like things that are floating around in your
Yeah,
serum, right? Antibodies get into your cells, but T cells could recognize sort of misbehaving, maybe compromised cells, right, and take some sort of action against them. That's the cytotoxic function.
yeah, and that isn't affected, but specifically by the spike antigen. No, I mean differences, but in different parts. So the T cell kind of reacts to different small chunks of the protein that are presented on like MH on a different protein. So
Yeah.
so actually mutations may not have the same kind of effect on the T cell response. Um,
Sure. Yeah. Yeah. I mean, that's kind of interesting. Again, like it just highlights how much we don't know mechanistically about how the vaccine works, right? Like we see the clinical endpoints,
mm,
right, when we're developing vaccines, but we don't know the mechanistic part of it because maybe it's that, yes, there's neutralizing antibodies, but it's also increasing this like T cell compartment or something, and that's leading to the immune function. So,
yeah, and the biggest biggest like vaccines are all ones that are almost there to prime the T cell response. So Moderna, they're all the DNA vaccines. They go in there, they make your cells produce viral protein, which is designed to annoy T cells. Same with the
mhm.
Chadox. So I feel uh, like that. Even if we see like antibodies that don't necessarily neutralize the spike protein, that doesn't necessarily mean that there isn't going to be an immune response because the antibodies are just one part of a much bigger system. So
Yeah.
I really want to see some hope from the T cell response. Um,
Sure, yeah. I mean, it's almost like, in some ways, I feel like what we might be saying is that if we see mechanistic information on how these vaccines work, and we see that they do rely heavily on the T cell response, we might feel more comfortable knowing that even if there's escape mutants of the virus, right, from antibody recognition, they may still be picked up, right, by their other antigens still may be picked up by those T cells. It would be nice to know what antigens those T cells that are anti-virus recognize.
yeah, and so also the next paper is touching on the topics we talked about now about like how cell organisms can escape from vaccines. This goes back historically to uh, global population structure and evolution of Bordetella pertussis and their relationship with vaccination. So
Yeah. Yeah. This one I like had this paper in my collection from a really long time ago, and I was just like I came upon like an article like an old Quanta article talking about how like things evolve away from vaccines, and it reminded me, I'm like, yeah, there's already research about this, right? Like it's not surprising to scientists to think about pathogens evolving away from vaccines, right? Like in nature, it happens all the time, right? Like through evolution, there's the host pathogen interactions, right? Like things get strong, and then things try to get stronger to break in and access their food source, I guess,
yeah,
their replication source. So yeah, this is like an interesting study that I read a really long time ago. It might be interesting to read again and just revisit in light of this stuff to maybe understand some of the dynamics here. But there are a lot of different vaccines that are made for Bordetella
yeah,
pertussis, and they're not always to the same antigen. And so I guess and since they've been using these different vaccines for like a long amount of some amount of time, you can see how the strains have responded, I
yeah.
guess, to those different vaccines.
So yeah, it's the bacteria. So am I right in saying it's the one that causes whooping cough?
Whooping cough.
Yeah,
Yep, whooping cough.
yeah,
It's part of like a childhood vaccination strategy in most countries.
right, yeah. So from what I've read, it was introduced in the 1950s, and then there was a resurgence in disease in the 1990s in areas of like high vaccination. So this study is like looks at the evolutionary relationships between a bunch of different strains, focusing on genes that are coding for vaccine components. So so if
Hmm.
it was found in the vaccine
Hmm.
and found in like the gene, they looked at like how much like those genes mutated to like be away from what was shown in the vaccine. Um,
Yeah, yeah. And I think the other reason why I thought it might be an interesting read is that there's that idea of I think they vaccinate against the toxin, but they also vaccinate against like an attachment factor.
right.
And it's like really like choosing different targets to vaccinate against cause different effects in the population. Because like you can imagine if I mean this is not possible, I think, with the virus to vaccinate against some sort of toxin. They don't make anything.
No.
Um, yeah, so like you don't really get the same we don't have that flexibility, but I think it's just for interest's sake, like for people trying to understand like what are vaccines exactly and like how do we deploy them? How could they be deployed? Like this is kind of interesting. How does it affect things in the long
Mm.
term? Because we might be settling into a long-term existence with coronavirus. Maybe this is interesting context.
Yeah. Right. Antibody neutralization of SARS-CoV-2 through ACE2 receptor mimicry. Um, so
Yeah,
yeah,
this is a strange one, right? Like,
yeah.
because you don't think about antibodies like directly binding the receptor site, but this paper reports some antibodies that do that. Um, and I think that's pretty interesting. I don't know how they made the antibodies in
So
here.
they basically, I think they isolated like a number of monoclonal antibodies from SARS-CoV-2 infected individuals, and they took the top three most potent antibodies, and they looked at them for like an in-depth structural and functional analysis.
Some of them did this, where they bind the receptor binding domain, the binding interface.
Yeah, and I mean, in the context of like what we've been talking about, this would be quite an interesting thing to look at to see how this monoclonal will react to these new versions of the spike protein that are evolving. Because theoretically,
Yeah.
if it is a good mimic of ACE2, then there's no way for SARS to mutate away from that without becoming worse at infecting.
It's like a really interesting, like we talked about the ACE2 decoys
Mm.
earlier, right? And it's like another way of making a decoy basically, where you have a antibody that hits this very specific interface. And if you remember, like one of the advantages of those decoys is that because like the spike protein's supposed to have this functional ability, right, to bind the receptor, it would mean mutation away from that is also mutating away, right, its ability to get into regular cells. So evading the decoy or evading this type of antibody is not helpful.
Yeah.
There's no evolutionary pressure to do that.
Yeah, so there's quite a lot of good stuff in this paper. Moving on to
I've never heard of this too. It's weird to me, like to think like, oh, like let's find an antibody that does this. But I guess that's one of the powers of monoclonal work is
yeah,
that your body makes a whole bunch of stuff and you just dig for the stuff that works.
yeah. Early induction of functional SARS-CoV-2 specific T cells associates with rapid viral clearance and mild disease in COVID-19 patients. So this is
Huh.
in. So I guess this links into what I was talking about earlier when I was talking about how T cells are really. I feel like T cells are really important for the COVID antiviral response, and I feel this paper is the reason why I have that opinion because this even though they shouldn't, because this was only done in 12 patients, but this is the thing that stood out to me was they did a longitudinal like study. So they took these patients and took regular blood samples where they quantified the viral RNA, they quantified the circulating T cells, and the antibody response as well. So they did all these three things at like time points, so you can actually see how this infection is developing in these 12 patients in quite a lot of detail. Um,
Hmm.
and
Yeah, I mean those are great data sets, right? Human data sets followed over a long period of time.
Yeah, so for each one, you're following one infection, and you know like how much virus there is and how that reacts to, say, if there's more T cells or if there's more antibody responses. And what they find is that so in cases that turned out to be less symptomatic, in cases that turned out to be less severe, it was characterized by an immune response where there's like lots of activated T cells early on, and
Hmm.
things that had a worse immune response tended to have more of a chaotic like antibody response, which again I guess ties back to long ago when we were looking at that memory B cells paper where the SARS-CoV-2
Mm.
drove B cells a bit mad to cause them to create a non-specific immune
Yeah.
response. Um, um,
So that's the same thing here, is that if you don't make a lot of how is that related? Tell me
oh,
again.
so that was basically related by the when they in more severe diseases they found that there was actually quite a lot of antibodies created, not necessarily specific
Gotcha.
antibodies, but lots of them there. Whereas in the less severe disease, there's many functional T cells um, early on. So that's the idea
Hmm.
that in order to have a good antibody response, a good immune response, T cells have to play a really important role. Um,
Hmm.
so
Yeah, it's interesting. I mean, I don't think we've seen the because it looks like they're actually looking at their T cell populations and seeing what antigen it ends up recognizing, right?
mm, yeah,
So that's interesting. Yeah, interesting information.
Yeah.
really characterizing like because they haven't split their population into severe and mild, you can get some association between what the profile should look like between the two, what's successful and unsuccessful.
Yeah, I mean, but obviously very small. They could easily have biased their sample somehow, and with 12 people, it's very easy
Mm,
to include something that could be completely out of nowhere. Um,
yeah,
so I want to make sure we keep that in context. But I found it quite interesting in the way that they did it and the way this model was done.
yeah, yeah.
Uh,
I think there's always a lot of respect when you can take observations in real humans and try to bring some meaning to that.
yeah. Speaking of which, direct activation of the alternative complement pathway by SARS-CoV-2 spike proteins is blocked by factor D inhibition.
Mm. Yeah, so this one's an older paper that I found published in October of 2020. But stuff is coming out all over the
Mm.
place. And I found it through, there's a new link in our Zotero. If you go to under selection and online collections, there's like a bunch of the places that we might visit to pick out papers. And there's a new one there now called COVID-19 Knowledge
Mm.
Map. And like there's tons of as everyone who listens to us should be familiar with, there's like tons of research coming out every week on this sort of stuff. How do you wade through it all? Well, this group with the knowledge map, they try to group things by categories, which is sort of what we're
Mm.
trying to do too. Group things by categories, but then they have like people who actually are working in those fields say like these are important papers for our understanding, right, of this phenomena. And so I picked this one out because I know we've been saying week after week that we haven't talked about a lot about the clotting part of the symptoms, or I guess terrible effects that
Mm.
happen upon infection, why you get blood thinners when you get infected. And so, yeah, this is one of those things that should bring some sort of mechanistic understanding since we're talking about um, blood proteins.
Yeah, and it talks about the complement system, which we haven't really covered that much, but that's like kind of an innate immune like version of the antibody response ish, where
Hmm.
and they look at like so it's like these proteins that are designed to like kill off viruses, bacteria by doing all these complicated chemical processes that allow it to recognize them. Um, it's, and yeah, it's also plays a factor in clotting as well. Um, yeah, so,
Yeah, so I mean, like systems are really closely tied, right,
mm,
in the body, and like the same, like right, clots are not bad things per se. Like they have some sort of adaptive ability, but it's like where that balance lies between the pathogens like involvement and your host response is like where the disease ends up sitting. So yeah, that's why I picked it because it's like, oh yeah, here's something where, right, like we can see like where is the balance with SARS-CoV-2 as it relates to the specific system? Could it be driving some negative consequences as well as protective ones? I don't actually know what they end up saying in this. I picked it out just like a little bit willy-nilly.
yeah. So yeah, they say that like
Oh,
inhibitor.
they're saying there's like some use of the spike protein in this.
Yeah, the binds like a clotting factor and helps, and that activates an alternative complement pathway on like some cell on host cells, which might. So I'm not sure what. Yeah, it's a bit involved. It's something that we could look into.
Yeah. I think they're Oh, I see. Yeah, complement killing. Yeah, because like it's like you can have things kill, but like it could also just like activate stuff,
Yeah. So,
and then that could be not good, right?
hmm,
Like if it activates these things on cell surfaces, you don't want them to be activated there. That could lead to bad things.
yes. Um, Speaking
Hmm. Okay.
oh, I was about to say speaking of bad things, but yeah, we've got another N501Y mutation paper, but this is interesting because it's a mouse study, and so it's a mouse study on. So the title is the N501Y mutation in SARS-CoV-2 spike leads to morbidity in obese and aged mice, and is neutralized by convalescent and post-vaccination human sera. So
Yeah.
there's a lot of stuff going on in this paper. I mean, so firstly, obese mouse models where you take mice and you feed them on a high-fat diet. So and then later
Hmm.
they do glucose tolerance tests to see whether they've have become diabetic or not. Then they've got the N501Y mutation, which at first I thought, oh, maybe they're interested in B117. No, this is a mouse adapted virus. So kind of similar
Yeah.
to the mouse adapted virus that we talked about a long time ago. But this one they decided to
Hmm.
do another one out from scratch, and I I feel like now,
Oh, they made another one from
yeah,
scratch.
they
That's
made another one, and this is just giving me so much anxiety because it's a gain of function experiments are usually very controversial, especially when you're dealing with like a pandemic strain. I remember like a couple years ago they're doing one on the flu virus in ferrets that
yeah,
was
fair.
really big. People were very worried about that, and now
Yeah.
people are doing this kind of gain of function stuff in mice, and it just blows my mind that, especially because it gets the N501Y mutation, which is now we now know is a big thing for human thing
Yeah,
as
it's
well.
not such a great mutation in the human population to have either. Yeah.
Yeah, so I'm just so part of me is like I put this up here to wag a finger and be like, please, when you do this, like let's know that taking safety precautions when you're doing this kind of research, or like that you've. But yeah, they
I
basically.
mean, it's interesting. I wonder what the rationale is for redoing the mouse adaptation, right? If there are these tools that researchers can share, presumably, um, why not get them? Maybe they were working on them at the same time.
Maybe
It was published before,
I,
yeah.
I I guess so. I mean, this is um. So it seems like, but again, like them doing it again is yeah. It doesn't like help me in like this paper more, but it's they still did it the first time, and and
Right.
that kind of went by with nobody saying a thing, and I'm like, okay, that's a bit odd. Um, but then there's some
Yeah.
like actual interesting stuff here. They look at obesity and age. So these are two populations in humans that have been found to might have some vulnerability to SARS-CoV-2, and then they also
Mm.
check with like convalescent post-vaccination human sera. So again, this is kind of interesting because now that we know N501Y is an important thing, knowing that human serum that was probably raised against a different strain might potentially have neutralizing activity for it. Um, so there's some stuff here.
I don't know. I When I read the title, this is also a preprint,
Yeah,
right?
it's a preprint.
medRxiv. Yeah, it actually it confuses me to read like the title of this paper because I understand this idea of like, oh, maybe they want to figure out if obesity, like what the factors in obesity, right, and diabetes might be that contributes to worse
Mm.
disease. But like I don't understand like in the intro, then they try to connect it to the variant, but like that doesn't make any sense to me. Like I don't know if we're learning anything about the variant in this, right?
I
Like,
don't.
yeah, the idea is that this, like we've seen it before, this mutation exists in mouse adapted strains.
Yes.
Um, so like really what we're just you have to just remember the model is a mouse getting sick.
Yes.
And like that's very specific, right? Mice getting sick, not humans getting sick. And so if the mice have diabetes, which mimics human diabetes, then maybe we have something to say about diabetes and getting
Mm.
sick. But nothing inherent about the virus itself.
Yeah, because I think a point that I think I need to raise is that normal mice aren't susceptible to coronavirus. They need to have these mutations in order
Yeah,
to become susceptible.
yeah.
And so this whole
Either the mouse has to change or the virus has to
yeah,
change.
and a lot of the other studies they change the mouse, and in some studies like this they change the virus, which again I don't think that says a much. So if they use like a human adapted mouse and then had this virus and they showed a different kind of disease etiology, then that would say something about the strain.
Yeah. If the mouse had the human ACE2 and then they used the changed virus on
And
it
they
and
compared
something else happened.
and they compared it with the
Yeah,
original, and then we'd be able to say something
yeah.
about the strain. This feels like they had a mouse adapted virus that they want to study obesity and age, and then they saw that this mutation was hitting the headlines and thinking, let's clickbait a few people. That's
I feel that very strongly. Again, that comes from reading. I'm confused. Like what are they trying to say exactly with the data? What's the story? And it's like a little bit too like navigated for the zeitgeist, I guess.
yeah. I think there might be some good stuff here, but I think it's like might have
I
been
think the data seems interesting.
yeah.
I would love to dig into this data. Yeah, the story, I'm not
Yeah,
sure.
the story I
Questionable
feel has been
story.
changed somewhat by external factors,
Yeah,
and
yeah,
that's something that
yeah.
we always have to be kind of aware of. Like there are trends in science that come and go. I mean,
Mhm.
sometimes we see papers that wouldn't have been created if wasn't some underlying trend. Um,
Yes, yes.
Yeah.
Okay.
So so now we're talking about I guess
Development and comparison of novel novel cross displacement amplification assays with other nucleic amplification methods for SARS-CoV-2 detection.
hmm.
I don't know anything about this MCDA assay.
Oh my God, me neither.
But apparently it's another way of amplifying things.
Yeah.
I've heard of LAMP. LAMP is like it's a type of isothermal amplification, so you don't have to thermocycle, and it uses like these like overlapping weird like hairpin style primers. So I guess there's another type that exists out there. I mean, I feel like choosing to read this would be something about learning about that particular technology. And they do side-by-side comparisons with LAMP and RT-PCR to see the sensitivity and maybe position this technology as something that could be good,
Yeah, I think
helpful.
the idea is like for diagnostics, these sorts of technologies rely on less like source, so they can be more sensitive because they've got because the isothermal means that they can kind of bind things more effectively, maybe, and they also
I think the isothermal helps because you don't need a thermocycler to do them. It's like more for like widespread testing,
okay,
right? Like more point of care testing, maybe places that like don't have like the people to run PCR machines over and over again. Yeah,
and so that basically means they've got primers that can bind to like different regions of the like chromosome. Oh, sorry, the
I think so. Yeah.
and because of that, it can be more sensitive because it requires more tests, more binding events to happen before it comes up positive. So, um,
Mhm, mhm. Yeah, if we choose this one to read, I think that we'll have to bring up like whatever the first papers for MCDA are and just see like how are they using this? Where was this sort of developed? Um, but yeah, mostly maybe just to unless people are super interested in that technology, then this is just like, oh, people are still working on the diagnostics front, right? Like diagnostics is still a very important thing
yeah,
in our battle against the pandemic.
yeah, and we've talked a bit. I mean, talk about the CRISPR use of CRISPR. This is like another sort
Yeah.
of thing. This is again a nucleic acid application technology, but it could be promising. Could provide more details about things. Um,
Yeah. Tests more
but
widespread.
I think mostly all
Or a cool tech that this lab does that they trotted it out um, for
yeah,
SARS-CoV-2.
so I guess mostly it's all test our ability to understand PCR, the polymerase chain reaction, and how they've changed that to make it into a diagnostic test, and kind of all the logic that
Exactly.
puts it together. So
A little bit of clever molecular biology is this paper.
yeah, um, so I was speaking earlier about like so this the next paper is something that I'm quite excited about. So it's called Cell-Free DNA Tissues of Origin by Methylation Profiling reveals significant cell tissue and organ-specific injury related to COVID-19 severity. So this
Hmm.
is interesting because it's about epigenetic testing. So
Yeah.
so we talked about a lot about testing to see if you had COVID, but what about testing to check out the severity of the disease, seeing what the clinical outcomes are? So you have COVID, it's in your body, it's destroying some parts, some organs, but which organs is it destroying? So this study
Hmm.
looks at circulating DNA in the serum. So the idea is that when an organ or cell gets damaged, it releases a bunch of the DNA inside of it. Now that isn't necessarily something that is going to be very useful if you're just looking at the genetic code because all your cells have the same genetic code. But if you
Hmm.
look, the epigenetic code is something different. So in different organs, they only need certain parts of your genetic code to work. The other parts are like methylated, and so you can tell kind of where a DNA is coming from based on that methylation
Uh-huh.
pattern. So the idea here is that they take a blood sample, take all the DNA,
That's so interesting.
sequence the methylated parts of it, and use that fingerprint to
Yeah.
figure out which organs are currently being damaged. Um,
Huh. That's more than just SARS-CoV-2
Oh
technology,
yeah,
right? Like
I was
that's
about
that's
to
the like
say this is a lot like that what I was talking about opportunistic papers where this wouldn't have happened without that context. But this is feels like this was something that they were working on. It was like, do you know what? Let's look at it for SARS-CoV-2. Um,
yeah.
then that doesn't necessarily mean it's a bad paper because sometimes there are some very interesting things that are happening in SARS-CoV-2
Oh,
that can be on.
absolutely.
So, for instance, like what cells are being damaged? Are they can be vascular cells or because we can't really because humans aren't see-through. You can't like see where the SARS-CoV-2 is like causing an infection. So this is just another way.
And you don't want to biopsy
Oh
someone
yeah,
like several times to figure out what's going on.
yeah. I mean, you don't even want to do that with an animal because you could be dissecting a
Yeah.
mouse for an hour or like for like days if you try and look at isolate individual cells. This is a much more refined way of looking at that, and you can get even longitudinal readings from that. You can take sequential blood samples and see how the disease is developing. So this is
Yeah.
it's also kind of,
That's
but yeah, it's a very epigenetic paper. So it's not so it's like on the edge of microbiology where we're looking at human health, but how it's being affected by a virus.
yeah, exactly. This is almost like taking existing knowledge of human genetics and right, human genetics, and then using it as a type of diagnostic, right? With the understanding that during infection, some stuff is getting lysed
Yeah.
and thrown into the blood.
Uh,
Cool.
the next paper.
Household transmission of SARS-CoV-2 and risk factors for susceptibility and infectivity in Wuhan, a retrospective observational study.
Yeah, this is looking at households with confirmed cases of COVID-19. So they were observing the spread of COVID-19 in households. So looking at like
Hmm.
who's being infected, how quickly it gets people infected, and and this is kind of interesting. It's been hitting like a few news articles, but it hasn't been talked about that much. But they found that found some interesting things. So they looked and did some calculations, and they kind of say that from their data they predict that people are more infectious before they show symptoms, which
Hmm.
is interesting. They also found that children and adolescents are less susceptible to the disease, but when they do get it, they give it to lots more people. So it's the kind of the takeaway they take out is that we need to be very careful about planning a school reopening in context of SARS-CoV-2. So this is a very like retrospective cohort analysis. So it's an observational study. So there's lots of confounders that
Mhm.
we need to be aware of, and we also need to be aware of like how the researchers are analyzing the data. Um, but it's
Yeah,
still
but it's like a large, I guess like what their strength is that they have so much data.
yeah,
It's a pretty large data set.
27,101 households with 29,578 primary cases and
Yeah.
57,581 household contacts identified. So they've done. There's a lot of people. It's far cry from the 12 people I was talking about previously. This is
Yeah, right.
this is a
Interesting. And this is from very early in the outbreak,
yeah,
this story. Yeah, this data is being collected from December 2nd, 2019, April 18th, 2020.
yeah. I mean, for reference, that's the usual timescale we expect research to come out. So that's so
Yeah, absolutely. Yeah.
so yeah. All the
Okay. And the last thing we got on our
ooh
list
yes.
is preserve a voucher specimen! Exclamation point. The critical need for integrating natural history collections in infectious disease studies. Um, and this one's not really, I'm not sure if this is a review, this paper, but I think it just had a really interesting perspective. I don't know if you guys listen to like, the Brain Scoop. That's like a YouTube
Yes.
channel that like she basically lives in a museum
Oh
collection and
yes.
always talks about the value of museum collections, right? In that you have this historical view of natural samples that like span into the past. So when something new happens and you're like, does this always happen? Does it always happen like this? Are there patterns in the past that can help us understand what's going on now? You can go into a museum collection and you can pick out the samples that you need and try to build a hypothesis. So this particular mini review is arguing for sort of the needing to store the microbiology element of every sample that's collected, right? Like
Yeah.
we should be also getting the gut contents and freezing that down for gut microbiota and like swabbing surfaces and getting the RNA at least, right? Which is what we end up using to identify the rRNA sequences to identify microbes to get some sort of sense of what's going on.
Yeah, so this is interesting because usually when we discover a new strain, we do like isolate that specific strain, and it either can sit
Mm-hmm.
it can sit in the researcher's lab for ages. So I remember in my old lab we had some strains going back to the 1920s or even earlier, just that have been
Yep.
passed on from like previous labs, and they've been kept like contiguously through like hospital collections. So sometimes
Yeah.
when you see a paper, you'll see this strain was a kind gift from X and X author. That's usually because they've got their own internal collections. But there are also some more public collections, like the American Type Cultural Collection, which stores
Yeah.
various strains at sites. So if you want to order a type strain, you can like pay them a little bit of money to keep them going, and then they will give you a version of that
Hmm.
strain.
Hmm.
But the interesting thing here is that these researchers aren't just like suggesting that; they're suggesting if you this is very much for zoonotics. So when you're
Yeah.
sampling strain, also taking samples of the host and the environment around it to get that context.
Hmm.
Um,
Hmm.
like for instance, when people are out take sampling for animals for zoonotic diseases, when they find an animal with a zoonotic disease, they'd like to take a sample from that animal as well, partly to make sure that
Yeah.
they've got the right species, because this is another issue. Because sometimes you get you get species are misidentified, or sometimes so having that whole specimen is quite useful if you're stu-
Totally.
and they make this like kind of point saying making the most of every sampling event. Uh,
Yes. Yes.
um,
Yeah, I think that's the big thing that they're saying here, right? Like it never hurts to have this more data. And here's this, and I think the other thing here's specifically, they basically go through all these examples of zoonotic infection, and they say this is how this information could help, right? Like this is where you would see this information playing a role.
Oh gosh, yeah, I remember like when the Zika virus was first discovered. There were only like 11 papers,
Yeah.
and I managed to read some
Hmm.
like all of them. And and the thing because one thing is one of these papers had the most brutal sentence I've ever read in any scientific paper. In fact, I think I've got it written down somewhere. I'll be back in one second to see if I can grab it. Ah, got it. Got it. Okay. It's so all right. Let me read to you the absolute worst thing that I've read in a scientific
It's
paper.
like in your personal notebook just because it was
It was
so
so
intense.
bad. I had to. I just had to. So, yeah, the paper was titled "Yellow Fever and Zika Virus Epizootics and Enzootics in Uganda." This was published in 1982, and they wrote after unsuccessful attempts to trap red-tailed monkeys in the Zika forest with the intention of bleeding and release, the monkeys had to be sampled by means of 12-ball shotguns. The
Oh
thing
my
that really
goodness.
gets me is that he said they wrote down specifically the intention of live bleeding and release, but they just couldn't be captured. So you can read the frustration of these researchers, and they're going like, right, fine, get the shotguns. These monkeys aren't coming on. It's like,
That's such a older papers type of like narrative too, right?
yeah,
Like it's really the research notes in some ways like made formal, right? Like a diary entry, like tried to live bleed monkeys, got frustrated,
yeah.
used
With the they
guns.
didn't even try. They intended to live bleed them, but they couldn't catch them. So it's like, ah, guys, these monkeys are they're like bad guys in a Disney movie. That's
But I like that it actually speaks to what we were talking about, right? Like it's very difficult to do these types of sampling things. So when they do it, it's good to get like a wide range of things, right? Like if those people were just studying monkeys, right, then they get monkey information. Let's get the viral information. It sounds like they were also looking for virus
yeah,
information
they're
then.
looking for virus. So this is part of the Ugandan like virus research institute, which was out looking for yellow fever viruses and other related things. So yellow fever lives in like the wild populations. So before you go get yellow fever outbreak, it usually appears in like monkeys and chimps. They try to sample them
Yeah.
first so they can predict when they're going to appear. So
Mhm.
that's a lot of like surveillance, and I think this paper is a lot about surveillance and that sort of thing of
Yes,
trying to
absolutely.
spot viruses and bacteria before they jump into human population.
Mm-hmm. Mm-hmm.
And yeah, having like an official voucher code for a voucher specimen, so that's on, so you can get not just the virus but also the context of where it's discovered. I think that's a very interesting idea,
Yeah.
and
Yeah, like linking up those two pieces of data essentially, right? Like knowing that this came from this biological context.
yeah,
Yeah.
I, so I think that
All right, we got to choose a
yeah,
paper,
we got to
right,
choose a
to
paper.
cover.
Okay, so
You seem kind of enamored by the cell-free
I I
methylation one.
really was, I really felt like that was that's mostly
It's not very
yeah,
pathogenesis
that's
though.
the
It's
thing.
very diagnostic.
Yeah,
Um,
so yeah, I think in terms of like
I like the. I mean, just from the mechanistic point of view, and because it was like a um, paper about like the disease itself, and we haven't talked about this clotting stuff or blood stuff, direct
Yeah,
activation of the alternate complement pathway. I'm interested in.
yeah, I'd be interested
Um,
in that. So yeah, I think trying to think of like because there are a couple of papers, but not really as much that you can really get your teeth into. Like so,
right.
because we've had like a couple of newsworthy papers come through, but they're very very short. Um,
Hmm.
so there's not much
Hmm.
we can get out of those. Uh,
I guess the induction of functional SARS-CoV-2 specific T cells associates with rapid viral clearance. I like that.
yeah,
Right? That's learned about in a small sample size, right? Trying to understand the T cell dynamics.
uh, yeah, I think that's good. I feel like the complement activating pathway is something that we haven't really explored very much. So I mean,
And then don't forget we also have all that's like, we still keep them in our potentials folder, stuff that seems interesting but we never got to. There's the in vivo secondary structure of the SARS-CoV-2 genome. There's NSP1, ORF8, olfactory transmucosal invasion. Um,
mm,
oh, inactivated spike protein revealed by cryo-EM. Yeah,
yeah. See, yeah,
super antigenic character of SARS-CoV-2 insert.
uh, right. I feel like of those, the Orphate, sorry, not the Orphate, the secondary structure of the SARS-CoV-2 genome. So I did a short video of that, of our discussion on that, and I feel like that's got a lot of interesting things that we could be talking about. Um, because
Yeah, I mean, that is interesting to me as well. I don't even know how you go about doing this comprehensive secondary structure analysis. That sounds interesting to
yeah,
me.
or we could try to figure out the PCR paper, um,
Hmm.
the paper on the multi cross disciplinary PCR.
Yeah, we already. I do think that because we did the CRISPR one,
Yeah,
I'm interested in doing something different.
yeah, true. Um,
So right now I'm feeling between three. It's the secondary structure, it's the complement pathway, and it's the T cells.
so of those, I feel like the complement pathway is the most different from anything we've done before.
Mm,
So
right, yeah,
yeah,
and it's a different like system. It's an important system in the body, blood.
yeah, it's an important system, and yeah, it's the one that I know the least about. So it's something that we can talk about a bit more because complement is there's there's a lot we can delve into with that. And
Yeah.
also
Yeah, I kind of like trying to round out our own knowledge and maybe like the type of knowledge that like we can point to people, right? Because like that's one of the things we've been doing. Like as we read things, we build the connections,
mm,
right, between different areas and we can be like, "Oh, yeah, that's really similar to that other thing that we read." And we've never really built the connections to this part of the pathogenesis.
yeah, because there's whole like this whole vascular story of that's happening with SARS-CoV-2 that we haven't really talked about like what part of the like I mean like how it's damaging the human body.
Yeah. Yeah. Yeah, I'm not sure if this is exactly that, but it might lead to thoughts around there. So
Yeah.
I'm for if you're up for it. It's an older paper, but again, I found it as recommended, right?
Mm.
Of something that's like something relatively recent that was published that people think is useful to understand this particular area. So
Yeah,
I'm
I feel like that because the secondary structure paper seems like to be really interesting, but it's also very much molecular biology. Whereas we really want to get back to pathogenesis. I think after we've done a few like things that are quite molecular biology. Uh,
sure. Yeah, absolutely. Let's understand some systems,
yeah, let's get back
some
to.
biological
I mean,
systems.
the stuff that is that yeah, understanding how the virus works, but isn't necessarily going to tell us how we because at the moment there's I mean, eventually we'll get more papers on like the new strains and what they're going to be doing. But at the moment, since we don't have that data, I think it's best to kind of focus on the kind of fundamentals of what CoV-2 is
Yeah,
doing.
I'm up for that. I'm up for that. Okay, so join us next week for our deep dive week where we're going to cover direct activation of the alternative complement pathway by SARS-CoV-2 spikes proteins is blocked by factor D inhibition.
Yeah, and we want to remind everyone that while we're very enthusiastic about microbiology and somewhat qualified, it is possible that we don't get everything right. So science is about thinking critically and asking the right questions. So if you have any questions, corrections, or comments, please let us know. And you can just drop down in the comments. All of our sources are in the comments. You can access our Zotero, and you can look at the papers that we've been looking at. Um,
Yeah,
yeah, and
I
feel free
totally
to reach
agree.
out.
Reach out to us over Twitter with the hashtag. We both believe that peer review is a process that we can all participate in. So we hope you all had a good time listening to us. If you follow us, subscribe for more microbiology content.
Yeah, it's been a pleasure chatting with you, Danny.
Same here, Fuzz.
Well, I'll tune in next week for more microbiology content. Where we'll nerd out about all things small that are great.